This is a brand new species of poison dart frog discovered in the Amazon. What's really, really cool about it is it is published in the journal PLOS One. I don't know, I was excited that it wasn't Zuki's. It's a nice journal where the whole shtick is, do you have something that is sound science? That's all they're looking for. Not how cool it is, just is it done well? And so this is a brand new, again, poison dart frog. It's found in the Brazilian Amazon rainforest. And what's really cool is that this is a mix of a lot of field work and then a lot of lab work as well. The reason I liked it is because it is in the Amazon. The way that they actually got to the location where the frogs are, and this is actually written out in the paper, y'all. They flew to a plain site in Brazil. There was then a 10-hour boat ride to a river basin. There was then a 10-mile hike with local guides, and it took them to an area that had palm trees, and there is where they found the frogs. And so at that time they took several samples, including adult frogs, tadpoles, and embryos, back to the lab. I thought it was incredible about how long of a trek they actually went on in order to find these frogs. Like, this was not just a nothing kind of trek, right? This was a very, very big deal to get. And what's even nicer, I think, is that they did some videography in this. They did some videoing of these animals too. So now we know a little bit about their natural habitat. They found a unique mating call that had 16 to 35 notes. And these calls lasted from anywhere from 8 to 16 milliseconds. They're really, really small, 15 millimeters in length. And it is the first new species of poison dart frog that was added to this particular genus for the last 10 years. So it's just a really cool way of finding this. The genus that this is part of is called the Ranitomea genus, and it's called Ranitomea aetherea. The aetherea is named that because the main trait that we see, and we'll take a look at the figures, is a long, almost sky blue looking line along the length of its body. There's also copper colored legs with some brown spots. And then the head is interesting because it is wider. The head is wider than it is long in terms of like the measurements of it, which is, I think it's really, really cool. They even noted other kinds of behaviors, right? So they actually watched the frogs and saw when they were active. And what they find is that they're more active in early and late parts of the day. But really interestingly, if it's raining, they change their behavior entirely to have higher levels of activity during the raining, which I thought was really, really interesting that they were able to do that. They also found that the frogs lived among the Bananaria brava plants and hide under fallen palm leaves. And these were the only location where these frogs were observed. So as the researchers climbed along that massive trail, like the river, the 10-hour river go, the 10-mile hike, there were no other instances of this frog, only when they got to this one particular site that they find it. That doesn't mean it's only located there. They do not have an estimate of how, or if any, endangered these animals are. It's just really interesting and cool science at this point in terms of what they know about it, in the geography. And they did some genetics too, Captain Krabs. Yeah, Captain Krabs. They did a little bit of genetics here, so they did DNA sequencing. All right, so a little bit more about this frog. New species of frog identified in the Amazon, and their trek was really, really cool. So in order to get to the site where they found the frog, there was a plane ride to a remote airport, like a small airport in Brazil. Then there was a 10-hour boat ride. Then there was a 10-mile hike with local guides, BT dubs. They didn't just go into the forest and do it. And then they found this frog. Now, all along that journey, they did not see any of these specimens. They don't know the range of this animal. They don't know how many there are, if it's endangered or not. But it's just really, really cool, hard-working science. Like, I really liked what they all went into in order to actually get to these particular sites. Yeah, right. So they needed the field guides in order to show them like that they've gotten and they're not lost. And so when we look at the figures, again, this is the journal PLOS One. So the whole shtick of this journal is a good science. So, Neil, that is a great question. Citizen science, Neil. There was, from the locals, here, let me actually show you. So here is the frog. There was talk of a blue frog with stripes running down its back. And that to the researchers sounded unique. And so they asked the locals, they're like, well, where is this? Where have you found this? And they were like, well, it's quite a journey. And so they had connections. Some of the locals met them at the airport, and then they went with them through the journey. And they had other support too, because when that 10-mile hike into the Amazon, right, if that goes wrong in any way, everyone's dead, right? You'll get lost. You'll never make it back out. Luckily for us, the Amazon is still big enough that people can get lost. And the only reason I say it lucky for us is because not so much has been chopped off yet, but it is like there's so much deforestation happening. The thing, Neil, that I find really interesting about this is that there's always suspicion that there's so much biodiversity in the Amazon that we don't know about. And so this is another example of like here in the Amazon, we have no idea about any of this, right? And now we find something brand new. And imagine if we put half as much effort as to what these scientists did in terms of their trek for a couple of other labs, how much more that we could probably find, right? It's just, there would be so much more, I feel like, that would be identified. Yeah, and the deep ocean too. Yep, in the same way. Their figures, y'all, are very, very beautiful. So this is the preserved holotype right here. So this is what they compare against, right? And then this is the in-life holotype. So look, like, this is what it really looks like. Tell me that's not a beautiful frog. Really, really unique in structure, unique spots, like the copper colored legs really pop in the living specimen, and then the blue stripes running down its back. Like, it is just really pretty, and it's still really small, right? This is a poison dart frog, so it's on the order of like 6 millimeters to 10 millimeters in length. Quite small. The other unique feature of it is that the width of his head is bigger than the length of his head. What functional reason, we don't know. We don't know what the functional reason of that is, but there you have it. Like, it is just really, really pretty. And again, it's this weird mix of this blue to then this like copper color limbs. Like, and just look at like the palette color really makes it look like it was a special handmade something, right? I love that delineation line between the body and the leg, right? To me, it then therefore looks like there's something really unique going on in their development, where in their developmental biology, there's this unique coloration that ends up happening, and that very, very fine delineation line. And that's not something very commonplace for it to be like this strong of a cut, but it's just really pretty. Here we have for both male and female. So we have in A, we have the male, and the female, and let's see, it's E, F, G. Yeah, here we go. So male, both sides, and we have female too. That is a great question, Corn, why they look like this. So these are poison dart frogs. And so a lot of the poison dart frog color hypothesis is more that it's a warning sign. So it's not that they need camouflage, it's their diet influences their poison state. So, which I think is really cool, by the way. I didn't know that as a kid. Maybe y'all knew this as kids. I didn't know about it. I just thought always poison dart frogs meant they were very toxic and will destroy you. It turns out it's very much based on their diet, right? So if you don't feed them the particular diet, then they won't be poisonous. They're told, like, so the ones in zoos are fed a different diet that they can be safely handled, and they don't have that toxicity to them, which is, again, a really unique feature of how diet influences that protection. That doesn't mean, Corn, that there can't be some functional reason for these colors on any level, like for camouflage, right? Because you are seeing this striping pattern, which is, that's another unique feature. Usually poison dart frogs, or most frogs, don't have this striping pattern. New ones aren't brightly colored. Yep, exactly. Although their underbelly spikes is a little bit brighter, like relatively speaking to like how much blue is on their dorsal side. On the ventral side, there's a fair bit of blue here. Yeah, Casey, I think they're beautiful, Casey. I love the stripes. Yeah, they're actually poisonous. Yeah, they're actually poisonous, Glued. If they were venomous, so Glued, for this delineation too, the venom, they don't have a venom gland, right? So because their toxicity is just associated with their diet. So because of the lack of the venom gland, that's another way that we know that they're not venomous. And that is a cheat code right there for this, but that dietary effect to it would make it be that kind of feature. Does that make sense? So Dr. A, they did capture, they got some for preservation. They got a preserved holotype. They also got a few living adults, a few living tadpoles, and embryos too. They noted that they were not able to successfully breed a colony, but they got a bunch of preserved specimens, at least at each developmental stage. Thought they poisoned their prey or something. So for these, Glued, they don't do any kind of like attacking of their prey. The poison for these, like, they'll eat like mealworms and whatnot, so they don't need to to actually attack their prey in any way. These animals, this poison is more for if there's a predator that tries to eat them. There are some videos that I will not show you today, Glued with Gold, but if you see a predator grab one of these frogs, it usually results in them spitting them right out. And more often than not, the frog is okay, at least from the videos that you can find. I'm sure there's instances where they swallow them in one gulp and the predator usually ends up dying as well. Here are, again, some more individual adult examples of both male and female just in the wild. Again, they got the nice images. We can see, again, the really nice delineation line. Casey, I think you and Melemony, Casey, will very appreciate this, I think. Look at that, like, strong delineation line between the limbs in terms of color and almost texture, like this metallic sheen to the limbs where it's like this copper color with spots, and then the rest of the body, which is this blue stripe, black stripe that they're going with. Yeah, so no sexual selection on these. So they didn't do any reproductive anatomy on this Smikes. But they did the imaging, at least on both male and female, and there wasn't a difference between the two of them. Which then it's like, well, then how do they do, attract members of the opposite sex? They did record some calls of how the animals do the croaking behavior. Now, it's not clear what these croaks are. Is it baiting call? Is it just general communication? Is it both? They weren't able to go into that depth. What the hope was bringing back some specimens to the lab that maybe they would be able to find out and delineate that, but they weren't able to, which is, again, an important note to make on here. Does the color intensify the more they eat the diet? If they stop eating the particular toxin, does the skin dull? So not for this one, or Jay. So the ones that have been tested for that, like, so the ones that are kept in zoos, it looks like there's no difference in color intensity. So it's not like, let's say, the flamingos, where if you don't eat the krill, then you won't have that pretty pink color. For these animals, it seems like you get that same bright color regardless of if you eat the diet or not. And what's neat about that is, if you take Tui, let's say, to the zoo, and y'all look and see in the poison dart exhibit, you'll still see some beautifully, really bright yellow colored frogs, right? Or other kinds of bright creatures. And they're not on this toxic diet. If you put them on the toxic diet, their color won't change. And it's also not developmentally set, it seems like. So it's, if you take them off the diet or put them on the diet at certain ages, it doesn't change things. It is an assumption and a half, Jay, to say that it is going to be the same thing for these animals. It's the conservative thing to say, I think, is that yes, it would probably be the same feature, but it was not tested, right? So when they took back the animals to the lab, they weren't able to maintain a population. So there could be. Yeah, so Jay, there is that does bring up the hypothesis of is that some kind of like bluff? However, if you give them the food choice of the diet that they're given in the zoo, let's say, versus the diet that they're given in the wild, and you get them do a behavioral choice, they will go for the food that gives them the toxicity. So I would suggest, to me, it's not necessarily faking it, because there is this innate behavioral preference that they're gonna go for. And it's interesting, it doesn't seem like there's a side effect to it for them, but it all could be one of those things too that isn't measured appropriately yet, right? In using geography to determine the species, in that future someone may challenge and ask where the science is. That's, yeah. So Neil, if they did have a map for this one, we can go ahead and jump to that. Here it is. So this is the geographical distribution of closely related species. And so Neil, this is that long river that they go on. And apparently, Neil, this was just, I think, a really cool story. They weren't able to go the whole 10 miles to this particular site, because apparently there's like some mountainous blockages in the path, if you will. So they had to disembark after 10 hours on this river. But yeah, this map is really cool, because they went that 10 hours on the river, and then you don't see the stopping point here, but they did have to stop before they got to the blue site, which this site right here is where the frogs were identified, and they had to do this 10-mile hike. Like, it was like something like up and around to get to this particular location. And the other colors here are same genus, this Ranitomea, but different species that you're able to see here, and so where they've identified them. You also see measurements of altitude, high altitude frogs, mid altitude frogs, some low altitude frogs as well. I like this map a lot. It gives us a lot of information. They even have the nice inset for you to be able to recognize like where on the continent it's located. So just a neat data set to it. And then they did, here was this like this interesting take on the striping pattern. So they didn't, this I feel like is a future directions question, but they noted that this striping pattern did have individual to individual variance. And Neil, I would love to know if this is, how much of this is influenced by genetics, epigenetics, and or injury. What do I mean by that? When we look at something like this gap in the blue signal, it almost makes me think that there was some cell death that happened there. So instead of the cells being able to be linked together with the blues as they normally would be, if something happens to that population of cells and they can't connect, right, then you will have the cells not be able to finish, and then you won't be able to have the full line and strand there. So that I think could be in part environmental, but then like the thickness of the signal like these, that's, I would argue, is most likely genetic, right? So like H and C are clearly different, right, in terms of how much intensity there is in these two zones. Maybe it has to do with the temperature of the water they're in, and then it's genetic and epigenetic. Mrs. Spice! Thank you for the combo, Jay. There's just a lot of really cool genetics to be figured out with here, and that's what they could have done had they established a breeding population in the lab. You could have done some really cool like mating schemes, and in theory even made genetic lines where you have a box, right, and everyone in this box is this type of genetic look, everyone in this box a different kind of genetic look, and you can figure out what genes are controlling these things and if it is environmentally regulated, right? And then you might have a model for epigenetics and color formation. It's a lot of ifs, and I know Jay Spites raising his eyebrow at me, saying, well, Balint, what about this? Jay, you know dang well I love that. What about that too? But that's just like, I think the fun part to me is that there are all these very unique directions that we can go in, and just like unique hypotheses on what else could be going on as a function of these colors. Like I mentioned, they did do some call analysis, and so they actually compared the way, they have a suspicion of what the call is, meaning that is it reproductive behavior or not. That suspicion is based on them comparing the calls to known songs of the same genus but different species, right? So in the same genus, if they have a particular call that they know is a reproductive call, then they compared a call of the new species and they say, okay, well, the shape of the call looks very similar to one that we know is reproductive in nature, so we hypothesize that it's reproductive, which I think makes sense. Again, they maybe could have figured out in a more controlled environment, but it's kind of cool that we're able to get that information. So comparing these calls, courtship calls, and then just like regular advertisement calls that the female is also interested in. So they have the male and female. And then here they have tadpoles even too. That's a great question, Neil. I know they've got the DOI here. I think it's a supplement. Neil, what I did was, this is FNJJV where everything's deposited. And then they've got like, I guess, the very specific ones. The thing I don't like though, Neil, is that it doesn't say in the paper, this graph and this point is associated with this other thing. That's how you would normally do it. It's like this supplementary file is associated with this particular observation stage we're looking at. So this is at 26 days of development, 37, and then this is the mouth here as well that we can see eye cells developing. Really cool bit of detailing of what we're able to see. Again, just neat science. More images too on what it looks like in nature. But I also like this, y'all. Just as an aside, this is, remember I said that their final location where they got to had palm trees, and that's where they ended up identifying these frogs. So you have the palm trees there of where they found the frogs. You see where they naturally found the tadpoles, where they have the adult frogs as well. So it's kind of a little bit of everything together. And I just thought that was a cool bit of additional final bit of science to put in there. It's like, yes, they did all this really nice genetic work, but then afterwards, they also gave us images of what this animal looked like actually in nature. So they did the genetic sequencing, they did some structural information, they did developmental stages, they even did some behavior in this paper in describing this new species. And so I think it's super, super well done. Okay, yes, Jay, we now have an image. I don't know if this makes it any better, I don't know if this might make it actually worse, Jay, but there is the little legs of this hungry baby tadpole, and then Mr. Spites, here is the Disney equivalent. It's Ryan Reynolds, so it's fine. Is that how that works? Wincing, I had no idea that these frogs did so much. Now, this doesn't mean that the new species of the week also does this. However, it's what is called your conservative explanation, or it's more parsimonious, meaning that if one does it and all do it, that means that it was a common ancestor in which this behavior had evolved, and it was then maintained in all these subsequent species. Versus the alternative hypothesis is that it randomly evolved a bunch of independent times. It gets much more complicated if you suggest that. It's easier to say it evolved at this point, everyone has it, and maybe a couple lost it, which I think is it's beautiful, right, Wincing? So that is the first science news story of the week.